Hi, my name is Dr. Robert Groysman with COVIDinstitute.org, and I've been asked many times, should I be worried about DNA or mRNA becoming integrated into our own DNA, our own genome from COVID 19 and from the COVID vaccines? That's what I want to address today. I hope you watch the entire video because I have some important information at the very end. So, as I said, there's been quite a bit of concern about whether the vaccines or the COVID virus itself, its mRNA, can become integrated into our DNA genome. Luckily, we have several defense systems in place to prevent foreign DNA and mRNA from becoming incorporated into our genome. So let's talk about the virus itself. The coronavirus RNA, that's the genetic material inside the virus, codes for four structural proteins, including the spike, the membrane, the envelope, and the nucleocapsid. That's the part that holds the RNA safely and protects it. All viruses have to go through a life cycle, which is attaching to a cell, penetrating the cell, replicating inside the cell, maturing and self assembling, and being released out of the cell. The mechanism of action of an mRNA vaccine is very similar to a mechanism of an mRNA virus or virus infection. It works by having the mRNA be translated by the host cell into a protein, the protein self assemble. What's in the vaccines? Well, both Pfizer and Moderna have a nucleoside modified mRNA that encodes for the spike protein. And it's combined together with some fat and cholesterol in a way that it forms a capsid to protect it, to protect this mRNA. Novavax is a little different. It doesn't have mRNA, it's actually a recombinant protein of the spike protein. This is what the COVID virus looks like. Um, there's the spike protein, the nucleocapsid, which winds up the RNA genome, the membrane, which makes up the membrane, and the envelope, which also helps make the membrane stay stable. So, what is the first obstacle? And this applies to both mRNA vaccines and COVID SARS CoV 2. So, first, the DNA or mRNA must survive being in tissue and blood, our plasma. And the reason is because our immune system is going to destroy it. Macrophages are going to find it and destroy it. There's also circulating nucleases, which break down any naked or free DNA or RNA. So, in order to protect it, whether it's a virus or a vaccine, it must be encased in a phospholipid membrane. And this is called a capsid. This is what keeps it safe. Anything that's unprotected will be degraded within a few minutes by nucleases that are floating around in our plasma. Obstacle number two, it has to enter the cell. That means that the capsid has to attach and be taken inside the cytoplasm of the cell. So, this is the second obstacle that it must overcome. The modified RNA versus regular RNA. Modified RNA is designed from the vaccines to basically withstand being digested by the endosome and to be able to escape it without being digested by an RNase. Whereas regular RNA typically gets destroyed, but viruses have evolved strategies to avoid that so that they can make it over to the endoplasmic reticulum where they can be translated into proteins. Obstacle number three, it must survive the cytoplasm. So it enters through an endosome, but endosomes have enzymes that can dissolve it and digest it. And it has to be able to survive that and escape the endosome in order to make it inside the cytoplasm. Next, the cytoplasm is kind of like an obstacle course. There's a ton of small organelles in the way that would prevent it from moving from the edge of the cell to either the endoplasmic reticulum or the nucleus. Obstacle number 4, it has to be able to move. So this mRNA or DNA, in this case mRNA, needs to make its way from the edge of the cell to either the endoplasmic reticulum or nucleus, and there's actually an active process. It crawls by using proteins to grab on to microtubules or actin and form motor complexes, and basically crawls slowly towards where it needs to go. I know it's crazy, but it's true. Obstacle number 5. Well, for a virus to replicate, at least a DNA virus, it has to get inside the nucleus. And while this doesn't specifically apply to SARS-CoV-2, it does apply to the vaccine, and I'll explain in a moment. It has to basically make its way through a nuclear pore and break through the nuclear envelope. Now, normally the nuclear envelope is shut down like Fort Knox. Nothing is getting in or out unless you're let in by the guards. So some viruses use their nuclear material to enter the nucleus during mitosis. That's when the nuclear envelope is dissolved and makes them way easier to get into a nuclear area where our genome is. Other viruses use something like a promoter to basically sneak in through a nuclear pore, even if the cell is not dividing. Now, let's talk about the virus and this COVID vaccine, at least the mRNA version of one. mRNA doesn't typically need to go inside the nucleus. It needs to go into the endoplasmic reticulum, where it's going to be processed by ribosomes to create proteins. However, our genome has a ton of reverse transcriptases built into our genome from prior virus infections going back millennia. These are from prior retroviruses. It was inserted into our genome, and we've kept this generation to generation. One particular one that's interesting is called the line 1. Now, COVID-19 does not have its own reverse transcriptase built into the virus. Some viruses do. But what this can do is it can transcribe the mRNA from an mRNA to a DNA fragment of the same type, same sequence. And because of this, a DNA fragment now has an opportunity to be inserted into our genome during mitosis, or if the DNA is being repaired. For instance, if there was a double strand break. The vaccine mRNA is non-replicating. That's very important. It does contain the spike protein, at least the 2 that we mentioned, but they also contain small DNA fragments that were originally used to create the mRNA fragments. Now, they were supposed to be removed, but instead they were cleaved into much smaller fragments and left in. This has been demonstrated in several samples that were studied. The mRNA from the vaccine contains these capsids. It's supposed to enter our cells, be translated by ribosomes into the spike protein or spike protein fragments that it codes for, and that is it. Neither the mRNA nor the DNA fragment integration into our genome was anticipated or expected. However, it is not so easy to integrate without an integration protein called integrase, which some DNA viruses bring with them. However, SARS-CoV-2 does not, and the vaccine does not have this either. So How easy is it to get into our genome? It's very difficult. It's very difficult to be incorporated. However, when you multiply the number of cells in our body by the number of fragments, it it may happen anyway. Is it harmful to have this fragment in our genome, and why? Um, Normally, our genes need to be activated by an outside a transcription factor in order for them to be unwrapped and transcribed into mRNA. We have about 10,000 genes, and we've studied about 6,000. And believe it or not, most of our genome is inert. It's basically junk. 98% of our genome, it's even actually slightly higher, is composed of junk, is composed of genetic material that is not used or utilized. All the genes that we normally use sits in about 2% of our um, genetic material in our genome, 2% of our genome. Um, so how can this be harmful? Well, it can be harmful if the fragment ends up going into a critical function gene or gets inserted into one and makes it either useless, non-functional, or it makes it hyperfunctional and can cause, for instance, something like cancer by speeding up the growth cycle, um, or be transcribed without a promoter or without an activation. It just becomes active on its own. So this is really the main issue. But considering we have 98% outside the bullseye, um, how many actually make it to the 2% to matter? This is what I wanted to point out, that even though this may happen, it may not be as significant as we think. Thank you. I hope you enjoyed this talk.